are found on the cell membrane. That's what we all know. So we are going to find that on the these receptors that are found on the cell membrane, they have got specific molecules that binds to them. So this molecule, it can be called a ligand molecule, and it is what we are referring to as a stimuli, and in this case it is an external stimuli. So when this binds there, this is an external stimuli or an external signal. When this external signal binds onto its receptor on the surface of the cell, this receptor will now transform that external signal into a functional intracellular response. So meaning that when this binds there, it is going to initiate a signal transduction that is going to cause this cell to react in a certain manner. So signal transduction, that is just the whole concept that is behind there. So as we have defined it, a process being in which a cell converts an external signal into a functional intracellular response. So now, as we talk about the same signaling and transduction signaling, there are actually types of signaling that we have to talk about and that we have to keep in mind and that we have to know. So we have got signaling that are under endocrine. So we can say endocrine signaling. So these endocrine signaling, these are just signaling in which a stimuli is released, and in this case we can say a hormone. That hormone is going to be released by a particular gland and it will travel through the bloodstream. So let's say that you're having this gland. This gland under endocrine signaling, it is going to release a hormone, and this hormone that it is going to release, it will move into the bloodstream and it will go onto its effector organ. And on the effector organ, as usual, the effector organ is going to have a receptor, and that receptor is what is going to receive this molecule and will bind it there. So endocrine signaling just involves the release of that particular external stimuli, which in this case is a hormone, that will travel through the bloodstream to a distinct target. Then we also have another one that is called paracrine signaling. So this paracrine signaling, it involves the signaling of molecules that would act on nearby cells. Let's say, for example, you are having cells like this. So you are going to find that these particular cells, as they have been arranged, this one will release a signal that is going to act on the neighboring cells. So this type of signaling is what is referred to as paracrine signaling. Then we also have another one which is known as autocrine, signaling. So this autocrine signaling, you just think of it like someone talking to himself. Yeah, that's what happens here. It's more like the cell is just talking to itself. In that manner, what I mean is that the signal will act on the same cell that has secreted it. So meaning that if this is a cell, it will release an external stimuli that will go and bind onto the same receptor on the surface of that particular cell that secreted it. Yeah, that is what autocrine signaling means. Then synaptic signaling just involves the transmission of synaptic signals. Let's say in the case whereby a neurotransmitter is being transmitted from one neuron to the other through the synaptic cleft or that synaptic junction there. So that type of signaling is what referred to as synaptic signaling. So these are some of the important signaling or types of signaling that would like you to know under this specific component. Then apart from that, what we have to know is that the same signal transduction actually has got messengers. These messengers are like those people that they send to deliver the particular information. For example, you write a note to someone, then you give someone, say, okay, I want you to take this note to this particular person. So the person that you are giving that note to deliver the note to, the person whom you wrote it to, that person is known as a messenger. The one who is delivering the information is known as a messenger. So now, what happens is that under signal transduction, we have what we call messenger molecules or messengers. So these messengers are the ones that are involved in the conveying of this particular information. So these messengers, we have got two types of messengers. We have got the first one, which is known as first messenger. So I've got the first messenger. Then we are also going to have the second messenger, obviously. The second messenger. So the first messenger, this one, how it works is that the first messenger is the same a molecule that is outside of the cell that will now convey the information into the cell by binding to a receptor. So what I mean is that this one, in this example that we had, this one is a ligand or an external stimuli that was coming from somewhere where it was being released. So this same one that was moving from somewhere where it was being released, coming to bind on the surface of this receptor, this one is known as the first messenger, which is an outside molecule that will convey the information to the cell by binding onto the receptor of that particular cell. So when this first messenger comes all the way from that particular village, it is being sent going to the town to deliver the information. It will come, and as it reaches the town there, it will now bind to the receptor. It will go to the specific house where it was sent. And when it gets to that house now, it will deliver the information, oh, you have been sent that, oh, groundnuts are ready, you can go and get them. So when it delivers this information in this particular town, then now people who are inside that cell, who are inside that house, will now start generating second messengers to say, oh, these now, they are going to start conveying the information to everybody in that house to say, oh, they have told us that it is ready, we should go and get. Yeah, so this is how the first messengers work. They are just those external signaling molecules that moves and conveys the information to the cell by binding on the receptor. Then the second messengers, as I've said, these are now going to be intracellular signaling molecules that will now be activated as a result of the binding of the first messenger. And these second messengers, as they are activated, they are going to relay and amplify that signal to initiate the response by that particular cell. So meaning that when this first messenger delivered that information by binding on the receptor, it caused the activation or the formation or the generation of these second messengers. When these second messengers are formed now, are the ones that are going to start now regulating, facilitating now the mechanisms and action inside that particular cell so that they can produce a response. And some types of these second messengers, we have types like cyclic AMP, which is known as cyclic adenosine monophosphate, also cyclic GMP. We are also having inositol triphosphate, also diacylglyceride. These are types of second messengers. So these are the ones that are generated upon the binding of the first messenger on the receptor. So now, having talked about second messengers, signaling types, and signal transduction definitions, let's now look at the types of signal transducers. So these signal transducers are just molecules that conveys an extracellular signal into an intracellular response. What I mean by signal transducers are just the same receptors that are found on the surface of the membrane are the signal transducers. So they are going to be conveying this information, which is converting this extracellular signal into an intracellular response via the action of the first messenger, as well as the formation or the generation of the second messenger. So we have got the first one, which we are all familiar with, which is the G protein coupled receptor. And by now, I believe that mechanism for this receptor is on our fingertips. So I've got one which is the G protein coupled receptor. So basically this receptor, how it does is that it will involve the binding of an external ligand molecule. So as we know, the cell will have what we call a G protein coupled receptor on its surface. So this surface will be connected, or it will be having a G protein molecule inside of the cell as it is embedded into that membrane of the cell. So when a molecule binds onto this receptor, what will happen is that this external ligand molecule that has bound into that particular receptor will cause now the activation of that intracellular mechanism that will cause the formation of the second messengers, and these second messengers will go further and activate enzymes inside of that particular cell, and these enzymes will now start generating other mechanisms inside that particular cell to bring about a response. So we are going to break it down further. Then the other type of receptors, we have what we call receptor tyrosine kinase. So this receptor tyrosine kinase is basically also a membrane receptor that has got an enzyme, a kinase enzyme on it. So as it is, we are going to have the membrane, and this is going to be a receptor. And now this receptor down here, it is going to have an enzyme, which is known as the kinase enzyme. And in this case, it is a tyrosine kinase enzyme. So what happens is that in this particular tyrosine kinase receptor, when a molecule binds onto this receptor, it is going to cause the activation of this enzyme, which is the tyrosine kinase enzyme. So this tyrosine kinase enzyme is activated via a process of autophosphorylation. So phosphorylation basically is just the addition of a phosphate molecule. So automatically there will be the addition of a phosphate molecule to this particular enzyme, causing it to be activated. And when this enzyme is activated, it is going to initiate intracellular signaling cascades inside of that particular cell, bringing about a response. So for example, let's say if it is insulin that had to bind there, when insulin binds there, it is going to activate this tyrosine kinase enzyme via the same autophosphorylation process, which this tyrosine kinase is going to cause the mechanisms inside that particular cell that will facilitate now the internalization of glucose. So that is how it is going to react. So we know that in a condition where having a stimuli, an external stimuli, which is insulin, the response that the cell gives it is internalization of glucose when that particular insulin binds onto the receptor. So this is what happens, and we get to know more about this signal transduction when we start looking at endocrine. Then the other one that we also have to know is the guanyl cyclase receptor, known as the guanyl cyclase receptor. So these guanyl cyclase receptor, basically what they do is that when a molecule just comes and binds to this particular receptor, this receptor will initiate a mechanism that will cause the formation of what we call the guanyl monophosphate. These cyclic guanyl monophosphates, we say that these are second messengers. When they are formed, they are going to cause further activation of other enzymes and so on and so forth, and bring about the response. So basically that is how this receptor works. Then the other receptor is the ligand-gated ion channel channels. These are channels, so they can be ligand or they can be voltage-gated ion receptors or channels. Let me just put channels here. So from the introduction that we had in nervous system, we will understand that these channels, they involve the binding of a particular molecule, which we can call, for example, let's say a neurotransmitter that can be excitatory or inhibitory. So when it binds to that particular channel, it will cause the opening of that channel, and it will allow now the efflux or the influx of ions, depending on what type of a molecule had to bind on that particular site. So we know that it is also under the same signal transducers, because it will be facilitating a reaction that will cause a response in that particular tissue where these ions are found. So it will just involve, it will open rather, it opens and closes in response to the concentration of a ligand molecule that has to bind onto it, or rather just the presence of that particular ligand molecule that binds on it, or due to the change in the membrane potential. We know that these that are voltage gated, we are going to find that when there is a change in the membrane potential, the resting membrane potential, they are going to be opening some, they are going to be closing in response to the changes in the membrane potential, basically. Then the other one, it is the nuclear receptors. So just from the word, the name itself, nuclear receptor, you know that this dude has to be found in the nucleus. So what happens is that basically it's not this receptor is found in the cytoplasm or the nucleus. So what they do is that this receptor being in the nucleus or just in the cytoplasm here, it means that they respond to molecules which are lipid soluble molecules, like steroid hormones. Yeah, so the lipid soluble hormones, or rather lipid soluble signaling molecules, are these molecules that are capable of diffusing through the membrane, because we know that the membrane, it is a lipid bilayer membrane. So meaning that it only facilitates the movement via diffusion through those molecules, rather those molecules that are lipid in nature are the one that tends to pass through there. So when you start doing endocrine, these are going to be a few of the concepts that you need to master you can either have a water soluble or a lipid soluble molecule. So what happens is that when that lipid soluble signaling molecule diffuses through, gets into the cytoplasm, it goes and binds to these nuclear receptors, and when it binds there, it actually regulates the expression of specific genes, or it causes the turning on of certain genes to cause the formation of proteins that are required in the body, or enzymes, or it tends to cause the inhibition of those particular genes, so that the enzymes that are being formed can be inhibited, or they can stop. Just like we talked about under regulation of enzymes, we talked about we have genetic regulation. So these are the receptors that are found under that particular regulation. So having talked about these, I would like to just give you an insight of how a G protein coupled receptor tends to react in a condition where a molecule binds onto it. So we have got this mechanism that is well drawn here and labeled, that is going to help us just to have a quick review of how this works. So what happens, as we said, that when a ligand molecule binds onto the receptor, which is a G protein coupled receptor, as you have seen, this G protein coupled receptor, it is connected to this molecule that is down, this complex here, which is having the beta subunit, also having the gamma here and the alpha, and the alpha is going to be connected with the GDP molecule. So what happens is that the binding of this ligand to this receptor, which is the G protein coupled receptor, it is going to cause the activation of this GTP, in which this GTP is going to be phosphorylated to GTP. So there's going to be an addition of a phosphate molecule. When that happens, this is going to become active and it will dissociate. It's more like this guy becomes rich and says, no, I'm going to leave you guys because we are not on the same level. You get the point, right? Yeah, so that's what happens here. So this guy is going to dissociate from this. I'll go my own way. So what happens is that this alpha and the GTP molecule that dissociates, that it is being activated, it goes and activates an enzyme that is known as adenylate cyclase enzyme. So that adenylate cyclase enzyme, when it is activated by this molecule, this enzyme basically will start converting ATP into cyclic AMP and OO. This cyclic AMP, we say that it is a second messenger. So this is how now the second messenger is going to be formed. When this second messenger is formed, which is the cyclic AMP in this case, it is going to go and activate protein kinase A enzyme, of which the protein kinase A enzyme in correlatoxin, we saw how it was working on those CFTR signal channels, and also inhibiting those other channels that were involved in the pumping of ions like that. This is how this guy works. Then on the other, it has got the ability to convert the phospholipase C, or to activate the phospholipase C, and this phospholipase C, when it is activated, it is going to start converting, are going to bring about a response. That's what will be happening basically. So as you have seen, we are going to have an external stimuli. This external stimuli can be any guy that will just be secreted, maybe let's say by the gland, and tells them, go on that cell. Inform the people from that particular town that there will be a war. So will come as a first messenger, binds there and shouts in that particular cell and say, there will be a war that will be coming. So this will act as an external stimuli. So when that guy does that, this guy will be activated and say, let me also inform the guys inside that we should prepare weapons. So it is going to do so by initiating a transduction. So meaning it is going to change that particular stimuli that came in and cause the formation of the second messengers, like the cyclic AMP, the inositide the inositotryphosphates, and the diacylglycerols. As these are formed as the second messengers, these are now going to tell these guys, the protein kinases and the phosphoinositide 3 kinase, to say, guys, there will be a war. So now these guys are going out to initiate the mechanism that will cause now this cell to respond to this particular external stimuli. So that's how this signal transduction basically works. So from all that information, it means that we have now understood what happens when a cell binds there, because we just basically used to say, no, when a cell binds here, there will be They say, oh, when the molecule binds here, they say, oh, become like this. But now we have known how does it become like that. This is what goes on behind the scenes. So basically, this is just a concept that we wanted you to understand on this particular topic, and questions to test how you have understood the concept will also be posted, followed by the document. Otherwise, if you have any questions, any queries, and any comments, we are free and we'll be very happy to help you. Otherwise, thank you very much for your time. Until next time, have a blessed evening.